review for the practical. Now, Keon has negotiated with me. He wants a multiple choice exam on the computer.
Who's recording? Who's recording?
So,
Me.
that is going
Keon,
to be
of course.
my goal. It is contingent on that I can get the room next door with all the computers in it. Okay?
Hopefully not.
So, you guys, if you want to bring your own computer.
Hold
then
on, that
that's
one based
fine.
off of Keon and not a vote from everyone else?
He said he was representing everybody. He said he was representing.
By a
everybody.
vote of hands, who is Keon representing?
For real?
Who is he not representing?
What does it mean?
University of Utah.
Okay.
It's with you.
Oh, okay.
That's a W.
Okay. She's not one of those. Okay. Remember. 2 acres.
Oh my God.
I'm gonna use it.
Right.
Oh my God.
All right, here we go.
You know how many more questions he adds on that? And they're just all super mixed up. I just think about what I should have studied and then what I didn't study. That much or that
Oh,
well.
what are you talking about?
Because you said on an exam, didn't he say that? He's like, I would make like 250 questions and then they're all mixed up. You just kind
Yeah,
of.
I'm good at that. I'm good at writing multiple choice questions. I hate them, but I like them. I mean, they're easy to write. That just means everybody gets a different test, which is fine.
Oh. Oh, okay.
Okay, here we go.
Got it.
So the
Turn the
mo-
light?
huh?
Turn the light? One?
Yeah, it's only one.
Sorry.
It's-
Oh, and then open up the closet.
Yeah, you can open the closet. Ooh, okay. Ooh, that looks really cool over there, that little light right behind the. Okay, so this is practical 3. I know it's just 2, but remember I moved things around. But this will be multiple choice. It's contingent on that it's going to be next door. So I will write a lot of questions. You guys will get 50 of them. Everybody will get a different one, a different exam. It's random. I'll randomize them. And so, you know, my multiple choice, as you know, do not normally just have 5 responses. They have more than that. So you just need to pick the best answer, but it's multiple choice to be right there in front of you. So we're gonna cover some of the tests that we did, the ones that are more challenging. I'm not gonna do a lot with the really easy ones, but the ones that are more challenging we'll look at here, so that we're all on the same page, so that everybody knows what the practical will be like, right?
You can add one on one of these, like, on one of the, labs that says this one has higher recording than all of them.
The nitrate test.
Yeah.
Yeah, it's my favorite one, that's why it has a lot.
Okay.
So, SIM, what does SIM stand for? What does the S stand for? Sulfur. Hydrogen sulfide or sulfur reduction, very good. What does the I stand for? No. And what does the M stand for? No good. How do you inoculate a SIM test? Straight, no, not stab the butt. It's a motility medium. So you're going to stab it with a straight needle, but about 3/4 of the way down. And then you wanna make sure that you don't move, okay? Now, when I put this exam together, I will be, hopefully I will be very clear about what you're looking at, but I will be in the room with you all and you can ask me questions about what am I looking at, okay? Because everybody's gonna have a different test. So if you look at these tubes, I want the results for them. So how will you report results for SIM? Now, hold on. What is the template? You write SIM and then you put a plus or minus after them, after the letters, right? Remember? So, on the first one, if you look at this, let me describe it for you. You have this growing here, but it's only growing at the site of inoculation, yes? So S, positive or negative? No?
Negative.
Negative. I, positive or negative? Negative. And M, positive or negative? So this would be S negative, I negative, M negative. Yes? Okay, let's do B. So here, you have red here, but you also have fuzzy. Now you can't really tell, but it's very diffused, but you have fuzziness going on in here, right? So S,
Positive.
No, S negative, there's no black. I, positive, there's red. And M, positive, because it's got diffuse growth. Now, I don't think I need to say much about this one, right? S, I, and M. Yeah. Now, remember, theoretically, we couldn't really tell motility from this because as hydrogen sulfide is produced, it diffuses out, and it then makes the entire butt black. But I told you all that for the sake of argument, that the organisms that we have selected are all modal, so therefore when you see a black diffused tube like this, you can immediately just say
Thank you.
it's H2S positive and modal. Okay?
So the M on A, that one was positive, right? The M on A was positive?
No, it was negative,
It was negative?
because I said it was only going to cybernetics.
They're all negative, right, on that one?
Uh-huh, for A, yeah, because what happened, Jalen, is when they put it in there, they did the little key on kick out, and then therefore they made it look like that.
So on B, it was, the S is negative, the I,
So
positive.
B
It's
is,
positive.
B is S negative, I positive, and M
Negative.
positive.
Oh, M
And I is
Endo.
representing.
Endo. Okay? Anybody else?
What was C?
C
Positive.
is positive, positive, positive. Okay, let me ask you a few more questions. What is the major substrate for the indole test?
The major substrate.
The major substrate?
Tryptophan.
Gobble, gobble, try,
Oh, tryptophan.
tryptophan, very
Yeah.
good, right, tryptophan, very good. What is the reagent for the indole test?
Kovac.
Kovac, very good. What is the presence absence indicator for hydrogen sulfide?
Say that again?
What is the presence absence indicator for hydrogen sulfide? The black, so what is the presence absence indicator?
The sulfur reduction?
That's the process. What is reacting with hydrogen sulfide to turn it black?
Is it the gas,
No, the gas is the end product, but what's in the media that's reacting with the gas so that it turns black?
Tryptophan?
Nope, that's
No,
only
no,
for.
it's what we put in it, right?
No, we don't put anything, it's already there. It's called ferrous ammonium sulfate, also known as iron. Oh, now we remember. I'm glad we reviewed this because I didn't want y'all to forget.
And what did you call that?
Ferrous ammonium.
sulfate, also known as iron.
Can we just say iron?
Well, that's gonna depend on what's on the multiple choice response. You gotta pick the best one.
Oh my god, so if there's sulfur and then iron, it would,
Huh?
if it had like the whole word and then it had iron, would you?
Hydrogen sulfide is the end product. Ferrous ammonium sulfate or iron is the reactant. It is the presence absence indicator.
Okay.
Justin,
Yes.
what was it?
No.
Anybody else? Okay, let's go on. Citrate, why is this known as a defined medium?
It's because, saturated is low carbon source.
That's true, but that's coming up in a minute. I'll point to you in a minute so you can answer that one. It's a defined medium because you know exactly how it's put together, remember? What is the indicator in the citrate test, the pH indicator? Bromoethyl blue. So remember, on this test, because we moved the decarboxylase and the deaminase to the fourth practical, that there's only two pH indicators. And that is phenol red or bromoethyl blue. Bromoethyl blue is only found in the citrate test. Any other test that I ask for the pH indicator, it should immediately be phenol red. Okay? At least for this practical. A positive test is which one? What color? Blue. And the pH of that particular is what? Basic, very good. So do not get confused when I ask for the pH. The pH is only acidic, basic, or neutral. Even though there might be other responses, it's only acidic, basic, or neutral.
So green bean.
Neutral. That's the way the media starts out.
So blue is basic?
Blue is basic. Yep. So now, Jalen,
Yes.
positive citrate means that the microbe, the organism could use something as a sole source of something.
Can you citrate as a sole carbon source?
Citrate is the sole carbon source, that's correct.
What does it say? It also says something about
Green to
ammonia
negative.
as
Green neutral.
Ammonium ion
Oh,
is the
yeah,
end
that's
product
negative.
and it changes the pH.
Okay.
So remember, when you release that ammonia, it increases the pH, therefore the pH of the tube goes up, becomes more basic. Okay?
Why did you say it's a defined meat?
Because we know exactly how it's put together in the recipe. You don't remember that
Oh,
from
yeah.
Unit 2?
What was to number 8, positive means that the microbe can use what as its sole source of?
Tell her
Citrate as a
to
source
in.
of carbon.
I'm sorry, I didn't catch
That was so
Citrate is the
fast.
sole source of
Sorry.
carbon. Citrate is, if you think about it, citrate is being used as the only nutrient. Okay? Questions? Alright, let's go on. MRVP. So first of all, I want to remind you all that if you know the name of the test, you know the name of the substrate, right? For instance, what is the substrate in the citrate test? So what is the substrate in the starch hydrolysis test? What is the substrate in the casein hydrolysis test? What is the substrate in the gelatin hydrolysis test? What is the substrate in the nitrate reduction test? See how easy this is? Why are you all worried?
The only one answered those
And
questions was
what is
Taylor.
it?
And you said, you see how easy that was?
The substrate. Substrate is what is being acted upon by the organism.
The
Right?
substrate is what is.
But you missed the most important point. If you know the name of the test, you know the name of the substrate. Now that is of the 14 tests you're working with this time, that's true for 10 of them. There's only 4 of them that are not like that. MR and VP are not like that. Methyl red and Volks-Kraskauer do not tell you what the substrate is. Therefore, what is the substrate in the MR-VP test? Glucose. Somebody said it. Oh, the Keishinator over there. Not hanging out with the Cheskinator today.
She's good.
Okay, so in the MR portion of the MRVP test, what reagent is added? Methyl red, very good. And if you get a positive test, what color is it? Red. And what does that indicate? What end products have been produced? No, thank you, Frank. Acidic, very good. Very good. Number 10, this test is part of the something battery test used in identifying something enteric bacteria. So what is the battery called? No, it's easy. MVIC.
Oh.
MVIC. MVIC stands for indole methyl red Volks-Kraskauer citrate. To identify what?
That's
Gram negative.
Gram negative, that's correct, amino. And then you guys already told me what the substrate was for the MRVP, which is glucose. Very good. VB. So, if the VP test is positive, the MR test would most likely be
Negative.
What does a positive VP test look like? Red, red. It's more of that A&M kind of burgundy color, right? What are the 2 reagents that are added to the VP test? Naphthol and? Potassium hydroxide, very good. And this test takes about 15 minutes, Tamisha, and I am very impatient. So I'm gonna add a catalyst. What is a catalyst? True, but what is it?
Well, I know it's called creatine.
Creatine is the answer. That's correct. Very good. Good. So.
Cheers.
Already?
like this stuff. So if you have an MR negative and a VP negative, what is the result?
Negative.
No, I think you're playing.
Neutral.
No, I think you're playing.
Positive?
No, I think you're playing.
Neutral?
Neutral. No, I think you're playing.
Ah.
I was like.
Error.
No acid, no neutral.
Yeah, you're right.
No acid, no neutral, therefore.
Basics.
Mhm.
Oh, wow.
If you got two positives, then what is it? Error. If you have positive MR, negative VP, what is it? Acidic. If you have negative MR, positive VP, what is it?
Acidic.
No, I think you're playing.
Neutral?
Neutral. I'm sure you know those. And I like to mix them up. I like y'all to have a little bit of charm in your life, you know.
I want to read my review.
Okay. Questions? All right, let's go on. Endo. Describe what a positive endo test looks like. It has a red light, very good. Indole is the end product of the digestion of which amino acid? Gobble gobble. Not turkey, tryptophan. Very good. Gobble gobble because it's in a turkey.
Yeah, so she said
But that's not right. It's not an amino acid. And the positive test is positive for which enzyme? Which enzyme breaks down tryptophan?
Indol's not on here.
Oh, no, where?
On here.
Where was I?
Oh.
Hello?
What did you say the amino acid was?
Tryptophan.
What enzyme?
Yeah, what's the enzyme that breaks down tryptophan? Hello?
Like
Add an A or C to the end of tryptophan, you get
tryptophan.
tryptophanase. Why is this hard? And what reagent is added to elucidate a positive or negative endo test? No, I think you're playing, not phenol red. Kovacs, yes, who said that? My bad. Yes, I didn't hear it. All I heard was phenol red from somebody over here. All right, the nitrate test. My favorite of all.
I hate it. So start that.
got it.
Okay. Just listen to me. I don't care about those. Just listen to me. In the nitrate reduction test, we're looking for the ability of the microorganism to reduce nitrate. So we inoculate, in 24 hours we come back, and we go to step one. In step one, we're looking to see number one, what is the substrate of the nitrate reduction test? Nitrate. Nitrate reductase is the enzyme. Okay, number two and number three, we add two reagents to step one of the nitrate reduction test. Please give me the names of those two reagents. Hmm? Naphthalene and sulfonilic acid. Very good. If we get a red color, does that indicate positive or negative? Positive for what?
Nitrate.
Nitrate does what?
Reduction.
Gets reduced to nitrite. NO3 to NO2. Be sure you know the difference. Okay? If you get no color, one of two things have happened. Either nitrate is still there in its pure form, or nitrate has been reduced to a gas. So we go to step two. And then step two, in order to figure out what happened, we add something. What do we add?
Zinc.
Zinc. And zinc's function is to do what?
Catalyst.
No, that can
Reduce. No?
Reduce. It's a reducing agent. Very good. I like the way Jalen is playing this game. She's just going down the line. So if you if you get a red color after add zinc, what does that indicate? Positive or negative?
Negative.
Negative. Very good. But if you get no color developed, that indicates positive or negative.
Positive.
For what?
Nitrate reduction.
To what?
Two nitrite.
No, negative weight.
Oh, to gas.
Absent.
To a gas.
Oh, gas.
And that's how easy it is. Chescanator, you love this stuff, don't you?
Love it.
Question. This is my favorite of all biochemical tests. Alright, easy one. The urease test. What is the substrate in the urease test? Urea. Very good. What color is positive? Pink or fuchsia. Very good. Why?
Because it's more alkaline.
True, because it becomes more alkaline. But what end product of the breakdown of urea caused the pH to go up? Ammonia, very good. Very good. Like it. Like it a lot. So what are the two end products in the breakdown of urea?
Ammonia and water.
No, ammonia and carbon dioxide. Very good. We don't care about carbon dioxide. It gets liberated into the environment, and who cares? But we do care about ammonia because that will change the pH of the tube. Will the pH of the tube, Miss Lakeisha, go up or go down? Up, because it becomes more basic. Right, so if you remember the pH scale, 7 is neutral.
Right.
Anything below 7 is acidic, anything above 7 is basic. Question. So do not be confused if I ask you, based on pH, what is the result of this tube? And I point to this, and you're gonna say what? Neutral. And if I point to that, it's gonna be base. Very good. Question. Just because it's a multiple choice question, do not think you should not study. Okay? Some people just think multiple choice are easier.
Not with 10 questions.
Let's go.
Right.
Alright, triple sugar iron. Name the 3 sugars in TSI. Hello.
Dextrose.
Oh, she loves that word dextrose. Okay, but lactose, dextrose, and?
Lactose.
And? Sucrose, very good. Now, Jalen, if you don't see dextrose on that, look for glucose. They're the same, okay?
Okay.
One of those sugars, glucose, lactose, or sucrose, is there in a lesser concentration. Which one is it?
Glucose?
Glucose, very good. What's the concentration? Yes?
Don't have that slide.
I don't either.
Oh, I might have added it later. I tell you what, before we leave today, you make sure I put it up. Fair enough? Okay? I change things because I feel like it's more helpful, and so sometimes I forget.
No, I don't like when you do that.
Oh. I'm trying to give you a better experience by changing them up a little bit, making them more presentable. All right, let me see. How does one inoculate a TSI test? And notice I put slant in there for a reason.
Slant, but
Streak the slant, stab the butt. SSSB. Okay? So remember, when we're gonna report the results for TSI, we're gonna draw 2 forward slashes. The first one is for the result of the slant. The second one is the result of the butt. And the last one is the result of gas. So check it out. I got yellow here, and I got orange here. Tell me, what is the result of the slant? Hello?
slant.
It's, yeah, well, it's acidic. So A, I didn't hear that. It's because you're saying it so low, you don't want me to hear. A, what about the butt?
There's
And, very good, and gas?
none.
None, no gas. Good? Nope, no, it's not negative, it's no gas. If you put negative, it's wrong,
No gas.
okay? Don't do it. Do not make my job hard. Okay, good. Here we go, another one, check it out. I got
Oh, that's pretty.
fuchsia here, fuchsia there. Slant. Slant. K, butt,
K for basic.
K. And what about gas?
K is alkaline.
Yep. What about the gas?
I don't see it.
No, well, okay, hold on, time. Don't make it that hard. I heard somebody I care about quite a bit say, I don't see none.
See, that's it.
The rule is that gas can only be produced in what conditions?
Acidic environment.
There you go.
In what environment?
Acidic. So if that thing isn't yellow, there can be no gas. Good? Questions? Here we go.
What was paint?
Alcohol, basic.
Oh, alright.
Alright, here we go. Check it out. So, what is added to the medium so that you can see the black color? Another way of saying that is, what is the indicator of hydrogen sulfide? And the answer is? No, I think you're playing.
No, is it not?
No, I think you're playing. Ferrous ammonium sulfate, also known as iron. Very good. Y'all are not having fun.
I'm
So the black color indicates the production of what? No, iron is the iron is the
Acidic?
presence absence indicator.
Oh, no.
Hydrogen sulfide gas.
Oh, okay.
Okay?
hydrogen sulfur.
So be sure when you read these things that you understand what's being asked for. Because if you answer quickly because you know the answer, a lot of times it might be not right. Okay, check it out here. I've got fuchsia here, hot pink, and I've got black there. Slant. What's the result? K, but A, very good, and gas. Hydrogen sulfide.
Positive, right?
Hydrogen, no, you can't say positive, you say hydrogen sulfide,
Wait, why is it hydrogen sulfide?
because it's black,
So we put cause it's hydrogen sulfide gas?
it's a gas.
Can you go to the last one real
The-
quick?
Which one?
The one before this one?
Yeah, so I've got fuchsia here, which is hot pink, and then I've got black there, right? And the Cheskinator's rule about gas is that gas can only be produced in acidic conditions. Oh, Angela's rule, okay. So, this is the slant, that's K. Here's the but, because it's black, that's a gas, and gas can only be produced in acidic conditions, even if you can't see it. It's A, and then you have hydrogen sulfide. Okay?
So we need to put like the actual gas that is being produced?
Correctamundo. It's either hydrogen sulfide, gas, or no gas. That's the answers.
Okay.
Good. Alright, last one. Here we go. This one here.
Wait, the one before.
What happened? What happened?
The one before real quick.
What?
The one before real quick. The slide before this one.
Oh. So I've got hot pink and hot pink. So K, no gas.
Yeah.
Okay? Oh. Alright, last one here. So I've got hot pink there, I've got yellow, but now I've got cracks and fissures down there. Got
Yeah.
cracks and fissures. So,
How do you see that?
K, because I'm gonna tell you.
Oh.
K.
Okay. Basic.
Oh,
It's basic.
I gave the answer. K, and then the butt, A, and then gas,
Hydrogen sulfide.
just gas.
You just said.
No, he said
It's not
black.
hydrogen sulfide, it's not black.
Oh.
It's not black.
Okay.
KK, no gas?
No gas.
No, there's.
Let's practice with some different ones. Here we go. Check them out. So let's do this one right here. So here I have hot pink, and I have yellow, and I have cracks and fissures. So here we go. Slant,
Okay.
butt,
A.
and
Yes.
and gas. Very good. Let's do this one. I got yellow here, yellow there. Slant, butt,
A.
gas. Very good. How about this one? I have hot pink here, hot pink there, but, I'm sorry, slant,
K.
butt,
A.
gas. Okay, good. Let's do this one. I got hot pink here and I got black there. Slant,
Okay.
butt,
Okay.
acidic. Very good. I was trying to trip you up, but you wouldn't fall for it, Cynthia. Very good. And gas. Hydrogen sulfide.
Cause it's black.
It's not. Retired.
It ain't hard.
I didn't know like what if I needed to put gas or like something gas.
So this is the TSI, right?
This is triple sugar iron. Yeah, TSI.
Full sugar iron.
You don't like that one?
Hydrogen.
Also known as H2S.
indicators in the name,
Indicators,
right? The indicators in the name.
That is correct, Amundo. A lot of times if you know the name, triple sugar iron, you kinda know what's going on, right? Well, you don't know the 3 sugars, but if you know there's 3 sugars, right? And then, okay, next. Oh, looky here. This is DNase test or DNA hydrolysis test. What's the subject?
DNA.
DNA. What is the presence absence indicator? Methyl green. Right, very good. Which organism, Staph aureus or Serratia marcescens, is positive? Serratia marcescens. Very good. Okay? Any questions? Starch hydrolysis. What's the substrate? Starch, very good. What is the reagent that's added?
Iodine?
Iodine, that's right. And which one, A or B, is negative?
A.
That's A is negative.
You said,
A is negative.
but on the PowerPoint.
I know, but I'm trying to trip you up.
Oh.
I wanna get your total understanding. I know you can read. Question. Cindy, don't look all depressed. I don't know what's going on there.
I'm sad now.
Why are you sad?
It's hard.
No, it's not. You're the one who made high grade last time. I'm not gonna fall for that.
That's not a guarantee.
Okay. So, gelatin hydrolysis. What is the substrate of the gelatin hydrolysis test? Very good. What is the purpose of placing the media after it's been incubated in the refrigerator at 4 degrees Celsius?
To see if it's gonna get like.
To see if it's gonna repolymerize or become solid again. Very good. So another way to say that is to see which tube is positive or negative. I like that too. Right? There's a lot of ways to say that. However, on a practical, there's only one right answer. You gotta
Oh,
pick it.
man.
Because it's gonna be multiple choice. I don't have it written yet. It'll be one of those. Okay.
Wait, what was
And
that?
which one is negative, A or B?
B.
B. Okay,
Because it's solid.
because it's solid. Any liquid is positive.
The enzyme like breaks down the
Yeah.
gelatin.
Cindy, don't be making faces like that. She's very
B is
demonstrative.
negative?
Okay, that's
Or
it.
wait, right? That's-
So I moved the
Oh, I missed it.
decarboxylates
I
and
didn't
the
write it down.
deaminase test to,
Yay.
those are also very kind of difficult to think about, but I moved them to the 4th practical. Okay?
And the 4th is our last.
Hey, I saw, will you turn on the lights real quick? Alright, so I'm not sure, is everybody okay with a multiple choice online test?
No. Sorry,
Keon, you'd be asking him questions the whole time anyway.
So how many people would prefer to do it the way I did it before?
On paper?
I don't want that way either.
Just throw a start and ask questions.
I don't like that way either.
You don't like, well, I don't have another choice, what other choice do you want?
Multiple choice?
It just kind of helped you because at the end you were able
On
to
a
like
paper.
reassure us.
Yeah.
I had like the
Yeah,
reassurance.
Like on a paper.
Not on the computer.
I can't do that.
I'm
'Cause I-
fine with
Because I
multiple
can't
choice.
put the
I
beautiful
don't care.
pictures
Oh,
anywhere.
that's right.
Like if I print it out on paper, Andy, it doesn't come out.
It's gonna be
like that.
gray.
And it's really important that you see colors on this thing.
I like the slides.
Oh, I see what you're talking about. Well, can't you just do the PowerPoint and put the picture and then multiple choice A, B,
No,
or C?
no.
Too much work?
No. It's either gonna be computer multiple choice or in person like that.
If the computer multiple choice though, like whenever you click to the next one, will let you go back to the previous.
Yeah, I'll let that happen, but, you know, it's gonna be randomized, and so I will probably come up, I'll probably come up with probably 200 questions and they'll be randomized. I have a lot of work. I wanna do what makes you happy.
That makes you happy too. More work.
Okay, let's discuss that.
Not the study group.
What would we do in its place?
Thanks to Misha.
Time for Thanksgiving?
Huh?
Time for Thanksgiving? We can bring a plate.
Oh yeah.
We'll bring
Every-
you tacos.
Everybody bring a dish.
Al pastor, 3 fries.
I appreciate
Ooh,
that,
3
but
pies.
that's not gonna influence me, okay? So y'all are not Kim Kardashian, right? Even she would influence me. So, I mean, I originally thought that, I thought y'all would be happy with a multiple choice online. Why are you not happy with that?
I'm fine with it.
Whatever.
Okay, that's what it's gonna be. That's contingent on if I get the room next door. Now, here's the other thing you can do. If you bring your own computer, you can take the test on your own computer. And that includes a tablet, but it can't be a Chromebook.
Hey, that's really rude.
Nothing works with a Chromebook.
They have it.
Yeah, there'll be a computer next door. I'm gonna go and I'm gonna ask for that room next door. Okay, but if you wanna bring your own computer, you can sit in this room all by yourself and you can do the test. You'll have 50 minutes to complete it.
How many
Okay?
questions?
50. 1 minute
50
per
questions for in 15
It takes
minutes?
me longer
It's
to
multiple
read.
choice. It's multiple choice.
50?
I read.
Hold on, you
Now,
said
hold on.
15?
That's like a minute per
50
question,
50
right?
questions in 50, 5-0.
Oh, okay, I thought you said 15 minutes
We
and
did
15
a
questions.
in-person test last time and we took 50 minutes. Why would it be different with multiple choice? It's easier, right? That's what you told me.
I think it'll be easier.
Yeah.
Multiple choice.
So we get 50 minutes?
50, 5-0.
60?
Huh?
What if we
Do you want 60 minutes?
Yeah.
Yes.
Okay, fine,
Less
done.
Just show me
pressure.
15.
Like,
Because we need time to think.
yes.
Sometimes
Oh my
more than a
God.
minute per
It's
question.
multiple
Well,
choice. I
it's
get to
multiple choice, but there's also like, you have, I mean, you know, some of those I'm like,
Sometimes you really have to look at the photo. You're like, okay.
what is
You
he
know.
asking you again?
Okay.
Think about it.
60. So let it be said, so let it be done.
60 minutes.
Done.
That's perfect.
Anybody have any other questions?
Yay.
You said extra credit for naming again.
No?
You said that.
It's practical for the last practical.
You said I'm gonna do it again.
Let me see what I can do. Let me see what I can do.
Oh yeah, the extra credit. That was awesome. If you ask me how to spell your name, I know how to spell it this
You're
time.
Mr. Extra Credit. I don't know why.
Add that extra R.
Yeah, so I'm thinking about allowing for people to do an unknown, but not to do it like in a traditional way. Because I feel like, because we were gone 3 weeks, that I just really haven't set you up for success that way. So, I know it's fun, but it's very complicated.
What, a project?
I don't know, what I would do is I would give you a tube that has an organism in it, and you'd have to figure out what it is. So it's like forensics. Oh, I never thought about that.
No, thank you.
Oh, my
no,
group.
no,
Why not?
don't you do it.
Yeah, it'll be
Don't
individual.
you do
Wait, so you're
it.
saying we're getting extra work?
But can't
No,
you
it's on the syllabus we've got, there's no extra work.
Instead of like.
Can't you give us like a map of like?
Why would I do that? I would, because that's what I want you to struggle with. I want you to
You don't want
struggle
us to look
with.
them up financial right there.
I would give you a table. You see that book behind Lakeisha? It's called Bergey's Manual for Determination of Bacteria. So I would give you, my version of that. It's in tables and you could use that.
If it was in groups, it'd be a lot easier.
Of course it would be.
Pairs at least?
Let me think about it. I don't even know if I'm gonna do it because it, I mean, if you think about it, our lab ends in about 3 weeks. And that doesn't give us any time. It doesn't give us any time. So the other thing I thought about is a one-on-one, you and me in a room, looking at each other. I'm asking questions, you answering questions back at me.
Oh,
Those are always successful.
But anxiety.
I have to look at paper and sit and think. You'll just distract me because
Why would I distract you?
Because I'll be like, boom, boom, am I gonna get this right? Like, I think there's more
I'm more interested
anxiety
in I'm more
there.
interested in how you think than what you know.